the chief question here is it safe and effective? And my answer to both of those questions is, I think so. Ultimately, the ultimate arbiter is going to be the FDA, but and of course the EMA. But I want to talk, sort of dive a little bit here into the specifics of why I think this is important. Relapsed refractory ALL, when we talk about the treatment of these patients, really we're talking about, a few sort of key therapies. Currently, we have blinatumomab, we have inotuzumab alone in combination, and then we have various chemotherapy combinations, or the integration of novel agents like bortezomib or venetoclax. When we look at all of those therapies, though, our remission durations are short, and largely dependent on a subsequent allogeneic transplant. And so that's sort of where we land. And when we talk about this particular therapy, we're actually talking about coming into exactly that space, right? So what happens when patients have exhausted their options, the blinatumomab or the inotuzumab or the transplant or whatever novel agents? What happens when we come into the space, and specifically what happens to come into the space for adults? We've tried with CAR T-cell therapy and ALL a few times, and again, it's been a bit of a rocky road. I think that Juno, or what used to be Juno now, BMS and Celgene, we tried with this, again, a CD28 based CAR T-cell therapy in ALL, but it was marked by these events of fatal cerebral edema, and it was challenging to really build beyond that. And I think that there were a lot of people who were really worried that this couldn't be safely done. We spent a tremendous amount of time in the phase 1 trying to figure out how to optimize it, not just the cell dose, actually. That was only one part of the equation, but actually thinking very critically about how to optimize the management of cytokine release syndrome and neurologic toxicities, and very specifically, this idea of early intervention. Could simply intervening earlier with steroids, with tocilizumab, an anti-interleukin-6 therapy, lead to a reduction in severe CRS and neurologic toxicity? The answer is yes, and this is what we had published in Blood, and we were very excited by that data. We really saw a significant decrease in these events, and when we looked at when we looked at attributable mortality to the KTEX19 product, it was low. Right. I mean, we saw an event of sepsis and in other words, we saw infection that was really more driven by the lymphodepletion, right, rather than by the CAR T-cell product itself. And when I say lymphodepletion, I have to sort of say that with a bit of a grain of salt, right? The patients have relapsed refractory leukemia. I mean, they're coming to us after having failed multiple lines with no meaningful bone marrow function, no ability to mount a good, immune response to pathogens. And now we're going to turn around and double down, right? We're going to give them bridging therapy to get them to the CAR T-cell therapy. Then we're going to give them lymphodepletion, and then we're going to infuse our CAR T-cells. So I think it's, when it comes to infection, we have to acknowledge that that's a reality of treating the ALL patient. But then when you really critically look at it, you say, okay, well, a couple of patients passed away from infection. I think you're also struck by the fact that, well, wow. I mean, that's actually lower than I thought it would be, having treated similar kinds of patients, in the sort of pre-CAR T, situation, right? When we had exhausted these same options and we were trying to come in with chemotherapy or, again, these novel agents. So, is it safe? Again, I'm going to talk specifically now about KTEX19 and say, again, I think so. We did see one event of cerebral edema. I want to qualify these cerebral edema cases because we think of them a lot as neurologic toxicity, and certainly that's how we classify them. But these are early events, and we have seen over the years, pulmonary edema. We've seen over the years, renal dysfunction. We've seen liver dysfunction, and all of these tend to occur, very early during the process of CAR T-cell immunotherapy, during that initial cytokine release syndrome phase. And, you know, when we think about sort of the rapidity with which these events emerge, quite honestly, you know, I think of this as almost more of a type of CRS phenomenon. And again, whether we call it CRS or neurotox is I guess, irrelevant, but it certainly speaks a little bit to, I think, the concept of what we're seeing when we talk about cerebral edema. And, you know, again, to further sort of, you know, dive a little deeper into this, it was the only case we only saw this one case across the entire study, including both the phase 1 and phase 2. And so when we look across all of these patients, we have to acknowledge this is also a very rare event. And I think that that's also important. I don't think anyone would, you know, would ignore it. I don't think we can or should. I think we have to acknowledge that there can be neurologic toxicity, but if we get, you know, sort of uber focused on that, then I think we lose sight of the bigger piece to this, which is, you know, again, thinking about risk not as just related to the product, but sort of what is the comparative risk. If we think about risk, what is the risk compared to other therapies we've given the same setting? And similarly, how do we think about risk as it relates to benefit? Meaning, is there a favorable benefit profile? And that leads me into the efficacy. You know, the efficacy on this trial was phenomenal. You know, over 70% complete remission with incomplete platelet recovery rate, so CR/CRi rate. This is really incredible for this group of patients. You know, I think very importantly, there was a high rate of true CRs. So it was almost 60% in terms of true complete remissions, and this really did translate into improvements in duration of response and overall survival, in relapse-free survival, sort of all the key surrogate endpoints that you would care about in a trial like this. I mean, median overall survival of 18.2 months for heavily pretreated adults with ALL. I think you can only step away from that and say, wow. And then if you focus on those who achieved a CR or CRi, it hasn't even been reached, and you're just sort of struck, and you say, okay, half of these patients are going to have a median survival that's not reached with, at least with the follow-up we have in the study, and that's incredible, right? I mean, over half, I'm sorry, 70% are gonna reach that, sort of, 16 months out, and we're still watching, and they're still alive. I mean, that to me is absolutely incredible. So this is with the FDA now, and actually, before I even say that, this is now published in the Lancet. So thanks to the Lancet for allowing us to share these phase 2 data. Is with the FDA now. I think the submission was in March or April, somewhere around that time frame. And so fingers crossed, hopefully we will be able to speak again in the future about a newly approved therapy for adults with acute